or cell wall using electron microscope, not general microscope. You can see by electron microscope. Cytoplasmic membrane is flexible, not strong, ruptures easily by some enzymes, by some antibiotics, by some antimicrobial agents. The cytoplasmic membrane can disrupt. With the exception of mycoplasma, bacterial cytoplasmic membranes lacks stroll. It means most of bacteria lack stroll, exception mycoplasma. Mycoplasma, a genus of bacteria, have a stroll in the cytoplasmic membrane, in their cytoplasmic membrane. Bilayer phospholipid. Bilayer phospholipid. Phospholipid molecules oriented so that hydrophilic means water-loving heads directed outward, and hydrophobic. Hydrophobic means water-hating tails directed inward. So, phospholipids in cytoplasmic membrane have hydrophilic and hydrophobic sections. Hydrophilic and hydrophobic sections. Proteins. Some proteins embedded in two layers of lipids. Bilayer lipids, lipid bilayer. And in cytoplasmic membrane, water can penetrate. Water can penetrate easily. And osmotic pressure created by cytoplasm. Osmotic pressure created by cytoplasm. This image, this shape is a structure of phospholipid in cell membrane, in cytoplasmic membrane. Yellow, polar, hydrophilic heads. Hydrophilic heads means like water. Hydrophilic heads. Its composition is phosphate group and glycerol. Phosphate group and glycerol is C. Contain enzymes to synthesis and transport. Contain enzymes to synthesis and transport, cell wall substance, and for metabolism and generation of ATP. The most role of cytoplasmic membrane is generation of ATP. D. Secret hydrolytic enzymes. Secret hydrolytic enzymes. VE. Regulate cell division. Regulate cell division. Mediation of chromosomal segregation during replication. Regulate cell division. Mediation of chromosomal segregation during replication. I will speak about the replication and the role of cytoplasmic membrane in replication because of mesosome decision in next slides. And cell motility. Cell motility. Before I said you about motility of bacteria by flagella. Yes, I said you Flagella originates from cytoplasmic membrane. Flagella originates from cytoplasmic membrane. Thus, cell membrane can relate it to cell motility. Function of the cytoplasmic membrane. Functions of the cytoplasmic membrane. Permeability barrier. Yes, permeability barrier for some materials can enter the cell, and some of them no. Permeability barrier means prevents leakage and functions as a gateway for transport of nutrients into and out of the cell. Some nutrients can transfer to cell, into the cell, and some nutrients no. It is permeability. It is because of permeability function of cytoplasmic membrane. Next, protein on core. Protein on core. Protein on core means site of many proteins involved in transport, bioenergetics, and chemotaxis. Some protein exist in cytoplasmic membrane and are involved in transporting proteins, and for chemotaxis, for example. And next, energy conservation, energy ATP conservation. Cytoplasmic membrane is site of generation and use of the proton motive force, PMF, the way of energy production, the way of ATP production in cytoplasmic membrane. Three types of transport molecules. Three ways. Uniport, symport, and antiport. Before I explain about these three types, it's better you see next slide. Yes, this is a schematic picture of transport, some ways of transport. At first, uniporter, one material from out to in red, uniporter. Symporter, two material in same direct, enter the cell. Symporter, synchronous, two materials synchronous in same direction can enter to the cell. But antiporter, two material, one enter, another exists. It's opposite of them. It's opposite. Anti means opposite. Red enter, green outside. Uniport, yes, this is uniport. Uniport means transports one solute at a time. Uniport means transports one solute at a time. Symport. Yes, symport. Two material. Symport transports the solute and co-transported solute at the same time in the same direction. Yes, pay attention. Two material at the same time in the same direction. In the same direction. And antiport. Antiport means transport solute in or out and co-transported solute the opposite direction. One material enter, one material exist in opposite direction. It means one material goes in, one material goes out. Yes, one material goes in, one material goes out. Antiport transports means one goes in, the other goes out, or vice versa. One goes in and one goes out. Movement of molecules through cytoplasmic membrane. Several ways for molecules to move through membrane. Several ways for molecules to move through membrane. 1. Simple diffusion. 1. Simple diffusion. 2. Osmosis. 3. Facilitated diffusion. Facilitated diffusion. 4. Active transport. I will explain about 4 ways of movement of molecules in cytoplasmic membrane. At first about simple diffusion, next osmosis, and next active transport. Simple diffusion. What's the meaning of simple diffusion? When a small nonpolar molecule passes through a lipid bilayer. Yes, simple diffusion means when a small nonpolar molecule passes through a lipid bilayer. Doesn't require expenditure of energy, don't need to energy. Yes, its transportation is very easily. No need any energy. Process by which some molecules move freely into and out of the cell. Process by which some molecules move freely into and out of the cell. And some molecules such as oxygen and carbon dioxide can diffuse from simple transport. It's for some molecules, carbon dioxide and oxygen. These materials are non-polar. Yes. Mechanism of diffusion. Diffusion is a movement of a molecule down a concentration gradient from an area of its high concentration to an area of its low concentration. Diffusion is the movement of a molecule down a concentration gradient from an area of its high concentration to an area of its low concentration. It means from higher concentration environment with high concentration to low concentration. Thus, it is easily transported and no need any energy. This process is passive. Yes, not active. This process is passive. It means it requires no input of additional energy. The concentration gradient alone is enough to drive the process. Types of diffusion. Two types of diffusion. One, simple. Two, facilitated. Yes, simple diffusion, facilitated diffusion. One, simple diffusion. In simple diffusion, molecules move directly across the membrane without the aid of carrier protein. There is no carrier protein. No need carrier protein. It's easily transportation. Simple diffusion is without the involving of carrier protein. For example, for oxygen and for carbon dioxide. For oxygen and for carbon dioxide. Small uncharged polar molecules such as urea. Small uncharged polar molecules such as urea. Yes, simple diffusion for oxygen, carbon dioxide, and urea. Two, facilitated diffusion. In facilitated diffusion, needs some proteins named carrier proteins. Movement occurs passively down a concentration gradient. However, the molecules require the help of carrier proteins to allow them to cross the lipid bilayer. Example, charged molecules, ions. Large uncharged polar molecules such as glucose. Charged molecules, ions, some ions, and large uncharged polar molecules such as glucose. And most of times, the facilitated diffusion is done for glucose. Facilitated diffusion, also known as facilitated transport or passive mediated transport. There are many definitions for facilitated diffusion, yes? Yes. Facilitated diffusion or facilitated transport or passive mediated transport is the process of spontaneous passive transport. Yes? Spontaneous passive transport as opposed to active transport. In active transport, we need energy, we need ATP to transport a material into the cell. But in simple diffusion, facilitated diffusion, no energy. Facilitated diffusion is a process of spontaneous passive transport as opposed to active transport of molecules or ions across a biological membrane, like cytoplasmic membrane, via a specific transmembrane integral proteins. Yes, some proteins involved in this phenomenon. Being passive, facilitated transport doesn't directly require chemical energy from ATP hydrolysis in the transport step itself. Rather, molecules and ions move down their concentration gradient, reflecting its diffusive nature. This picture, this image shows carrier protein. This is cell membrane, blue phospholipids, green proteins. Yes, this is protein channel, extracellular space, intracellular space. Some materials from extracellular space comes to intracellular space. What? We are carrier proteins. In facilitated diffusion. Ion channels for ions and for some materials. Three-dimension rendering of facilitated diffusion. You can see insoluble molecules in green, yes, in green color. In green color, you see insoluble materials, insoluble molecules. Insoluble molecules connect to receptor sites, and via carrier protein enter the inside of the cell. This is carrier protein in brown color, carrier protein, and green color is a material, a molecule. Transport proteins or transporters responsible for facilitated diffusion. What is facilitated diffusion? Facilitated diffusion is type of passive transport that is dependent on single transport protein carriers. Yes, with transport protein carriers and without energy. Transport proteins, rate of solute entry, carrier-mediated transport, and simple diffusion without carrier proteins. Just about active transport. Now about active transport. Active transport is very important. It needs ATP because of the difference in gradient concentration. A material from low concentration comes to high concentration, and thus it needs energy consumption. Active transport. Active transport is the movement of molecules across a cell membrane. Active transport is the movement of molecules across a cell membrane from a region of lower concentration to region of higher concentration. Yes, from low to high. but in facilitated diffusion from high to low. But here, from lower concentration to higher concentration region against the concentration gradient. Active transport requires cellular energy to achieve this movement. There are two types of active transport. Yes, there are two types of active transport: primary active transport and secondary active transport. Primary active transport and secondary active transport. Primary active transport that uses ATP, adenosine triphosphate, ATP, and secondary active transport uses an electrochemical gradients. Electrochemical gradients. This is the difference between secondary and primary active transport space. Unlike passive transport, which uses kinetic energy and natural entropy of molecules moving down a gradient, in active transport uses cellular energy. Yes, in active transport, cellular energy is needed to move them against a gradient, polar repulsion or other resistance. This is the difference between active transport and passive transport. Active transport is usually associated with accumulating high concentration of molecules that the cell needs, such as ions, glucose, and amino acids. Yes, some ions, some carbohydrates like glucose, and some amino acids transport via active transport. PEP group translocation. PEP means phosphoenol pyruvate, a material with high energy. Phosphoenol pyruvate. PEP group translocation is a way of transport for some carbohydrates, not anything, not H2O, not carbon dioxide, not urea, not anything, just for some carbohydrates. Just for some carbohydrates. Some carbohydrates and some bacteria can do PEP group translocation. And group translocation is a specific way for just some bacteria. It means eukaryotic cells, animal cells, plant cells, human cells, protozoa, and fungi don't have PEP group translocation. Yes, PEP group translocation is just for bacteria, some of bacteria and some of carbohydrates, not all of bacteria, not all of carbohydrates. Yes. PEP group translocation, also known as the phosphotransferase system, or PTS, is a distinct method used by bacteria. Yes, by bacteria, underline bacteria, for sugar uptake, where the source of energy is from phosphoenol pyruvate. It is known to be a multi-component system, or multi-component system that always involves enzymes of the plasma membrane and those in the cytoplasm. PTS or PEP group translocation. PTS or PEP group translocation. The PTS system is a type of active transport. It needs ATP. It means it needs ATP. The PTS system uses active transport. After the translocation across the membrane, the metabolites transported are modified. It is very complex. You can see in next slide the picture and the way of the sugar, for example, glucose transport in this way. The bacterial phosphoenolpyruvate sugar phosphotransferase system, PTS, transports and phosphorylates its sugar substrates in a single energy group step. This transport process is dependent on several cytoplasmic phosphorylate transfer proteins. What are the proteins? This include enzyme 1, 2-HPR, H means histidine protein, HPR. Next, enzyme 2A and enzyme 2B, as well as the integral membrane sugar. And integral membrane sugar permease 2C. The PTS enzyme 2 complexes are derived from independently evolving 4 PTS enzyme 2 complex superfamilies that include 4 superfamily. Yes, they include 1 glucose, 2 mannose, 3 ascorbate galactitol, and 4 DHA superfamilies. DHA or dihydroxyacetone. There are 4 superfamilies. PEP group translocation. This system, or the phosphotransferase system, is involved in transporting many sugars into bacteria. Yes, just for sugars and just for bacteria, including glucose, mannose, fructose, and cellulose. PTS sugars can differ between bacterial groups, mirroring the most suitable carbon sources available in the environment. Every group evolved. For example, in E. coli, Escherichia coli. Escherichia coli is a gram-negative bacteria. In Escherichia coli, there are 21 different transporters. It's very important. There are 21 different transporters, for example, 2C proteins, sometimes fused to 2A or 2B proteins, which determine import specificity. Yes, it is specific for a bacteria. For example, these proteins are specific for E. coli, for Escherichia coli. Of these, 7 belongs to a fructose. 7 case belong to fructose family. 7 belongs to glucose family. And 7 belongs to other PTS permease family. 21 transporter, 7, 7, 7. 7 for fructose, 7 for glucose, and 7 for other things. The phosphoryl group on PEP, phosphoryl group on phosphoenol pyruvate is eventually transferred to the imported sugar via several proteins. The phosphoryl group is transferred to the enzyme 1. Yes, the phosphoryl group is transferred to the enzyme 1, histidine protein, HPR. HPR is heat-stable protein. HPR is heat-stable protein. And enzyme E2. Enzyme E2 to a conserved histidine residue, whereas in the enzyme E2B, the phosphoryl group is usually transferred to a cysteine residue and rarely to a histidine. Sometimes histidine, sometimes cysteine. Cysteine, histidine. And in the process of glucose PTS transport, yes, glucose transport via PTS system, it is specific for enteric bacteria. Enteric bacteria like E. coli, Escherichia coli. Escherichia coli is an enteric bacteria. PEP transfers its phosphoryl to a histidine residue on E1. In next slide, you can see this explanation in E. coli and in Bacillus subtilis. PEP transfers its phosphoryl group to a histidine residue on E1. E1 in turn transferred the phosphate to HBr. HBr phosphoryl transferred to E2A. E2 is specific for glucose, and it further transferred phosphoryl group to juxtamembrane E2B. Finally, E2B phosphorylate glucose. E2B finally phosphorylate glucose as it crosses the plasma membrane through the transmembrane enzyme 2C. Enzyme 2C, and forming glucose 6-phosphate. Glucose converted to glucose 6-phosphate. The benefit of transforming glucose into glucose 6-phosphate is that it will not leak out of the cell. It's very important. It's very interesting. The benefit of this transportation is the glucose will not leak out of the cell, therefore providing a one-wave concentration gradient of glucose. The HBr is common to the phosphotransferase system of the other substrates mentioned earlier as in upstream E1. Yes. What I explain about PTS, you can see in this shape. For E. coli, Escherichia coli, and for Bacillus subtilis. At first, PEP, phosphoenol pyruvate, convert to pyruvate and release ATP, phosphate. And phosphate transferred and transferred and transferred, and finally glucose convert to glucose 6 phosphate. Yes, the enzyme are E1, E2, HPR protein, E2A, E2B, and 2C involved for this transporting. And about Bacillus subtilis 2, PEP, E1, HPR, E2 enzymes, E2A, E2B, and 2C. Glucose convert to glucose 6 phosphate. Another way of the transport of some material is osmosis. But osmosis is for water, not ions, not glucose, not urea, not organic material, not. Just for water. Yes? You know about different solutions. Isotonic solution, hypotonic and hypertonic. Hypotonic means low concentration. Hypertonic means high concentration. Water can cross from low concentration to high concentration environment. Yes. Thus, osmosis means movement of water across selectively permeable membrane driving a difference in solute concentrations on the two sides of membrane. Yes. Yes, cytoplasm. Cytoplasm. Excuse me, 5 minutes rest and I will continue. Cytoplasm have a solution. It's a solution include of some of materials, for example, sugars, amino acids, salts, dense gelatinous solution of sugars, amino acids, and salts. 70 to 80% water. Most of cytoplasm compositor, it's fluid, serves as solvent for materials used in all cell functions. Gel-like matrix of water. Gel-like matrix of water, enzymes, some nutrients, wastes, include organic wastes or inorganic wastes, and some gases, and contains cell structures like numerous ribosomes and polysomes. Yes. In cytoplasm, you can see water, some enzymes, hydrolytic enzymes, some nutrients, for example, inclusion bodies, and mesosome and ribosome. No ER or membrane-bound granules. No endoplasmic reticulum. No in bacteria. Endoplasmic reticulum is just for eukaryotic cell. And organelles in cytoplasm lack membrane in bacteria. Yes, in bacteria lack membrane. Granules or inclusions. Bacteria's way of storing nutrients. Some nutrients in bacteria are in storing form. Yes, storing form in cytoplasm. For example, glycogen, phosphate granules, sulfur granules. I explain about these granules decision in next slides. Staining of some granules aids in identification. Yes, we have some specific staining methods. For example, Albert staining or Neisser staining. Albert or Neisser staining is used to observe the granules of Corynebacteria. For example, Corynebacterium diphtheriae, cause of diphtheria. Corynebacterium diphtheriae bacilli have granules, some granules named metachromatic granules or volutin granules. It's related about phosphate inclusion. And some staining method is used to observe granules. And it is a type of identification item for bacteria. Inclusion bodies. All of bacteria have inclusion bodies in cytoplasm, but the type of inclusion bodies are different. Yes, they are non-living structures present in the cytoplasm. Inclusion bodies are non-living structures present in the cytoplasm. The inclusion bodies may occur freely inside the cytoplasm. For example, cyanophysin granules. Yes, cyanophysin granules in cyanobacteria. Volutin or phosphate granules. Volutin or phosphate granules in Corynebacterium. Corynebacterium diphtheriae. And glycogen granules. Glycogen granules in some Clostridia or some enteric bacteria, or covered by 2 to 4 nanometer thick non-lipids, non-unit protein membrane. For example, gas vacuole, carboxysome, and sulfur granules. On the basis of this nature, or of their nature. 3 types of inclusion body. 3 types of inclusion bodies. 1, 2, 3. A, B, C. A, gas vacuole. Yes. 3 types of inclusion bodies are A, gas vacuole. 2, or B, inorganic inclusions. Inorganic inclusions. And C, food reserve. Food reserve. In next slide, at first I speak about gas vacuole, and then inorganic inclusions, and finally about food reserve. A, gas vacuoles. Gas vacuoles. There are gas storing vacuoles found in cyanobacteria, purple and green bacteria, and a few other planktonic forms. Gas vacuole is seen in photosynthetic bacteria. Yes, cyanobacteria are oxygenic photosynthetic bacteria. Oxygenic photosynthetic bacteria. In bacteria, we have two forms of photosynthesis: oxygenic photosynthesis and non-oxygenic photosynthesis. Oxygenic photosynthesis in cyanobacteria, like green plants, like algae, and non-oxygenic photosynthesis is related to anaerobic bacteria, some anaerobic bacteria. For example, green sulfur bacteria or purple sulfur bacteria. These bacteria have gas vacuole. Yes, these bacteria have gas vacuole. Thus, gas vacuole are found in cyanobacteria, purple bacteria, purple sulfur bacteria, and green sulfur bacteria, and plant tonic forms of organism. A gas vacuole is without any covering of its own. It consists of a variable number of hexagonal, hollow, and cylindrical gas vesicle. Each gas vesicle is surrounded by a single non-unit, non-lipid protein membrane having ribs or folds. The membrane is impermeable, not permeable. Pay attention. The membrane is impermeable to water. Yes, impermeable to water, but is permeable to atmospheric gases. Is specific for gases, for some atmospheric gases. Gas vacuole is impermeable to water, but it's permeable to some gases in atmosphere. Gas vacuoles protects the bacteria from harmful radiations. It's very interesting. Gas vacuole protect bacteria from radiations. For example, UV radiation or gamma radiation. B. Inorganic inclusions. Inorganic inclusions. Several types of inorganic granules occur in bacteria. Several types of inorganic granules occur in bacteria. They include volatile granules. I spoke about the granules, yes? This include volatile granules. I said it's related to Corynebacterium diphtheriae, Corynebacteria. Volatile granules, sulfur granules. Sulfur granules found in green sulfur bacteria, purple sulfur bacteria. And iron granules. Iron granules found in iron oxidizing bacteria. For example, Gallionella, Caulobacter. And magnetic granules, etc. Magnetic granules, for example, Equispirillum. Because of the ability to pick up different colors with basic dyes, they are called metachromatic granules. Yes, because the ability to pick up different colors in different environment, different colors with basic dyes, they are called metachromatic granules. Two common types of inorganic granules are volatile granules and sulfur granules. Yes, the most important of granules are sulfur granules in photosynthetic bacteria and phosphate granules in Corynebacterium. Volutin granules, sometimes said Bob Ernest granules, Bob Ernest granules or volutin granules, are polymetaphosphates which function as a storage reserve of phosphate. It's related to phosphate storage. And sulfur granules occur in bacteria leaving a sulfur-rich medium, like the one which pick up hydrogen sulfide for obtaining reducing power in photosynthesis. Iron granules. Iron granules are similarly found in those bacteria which metabolize iron compounds for obtaining energy. Yes, iron granules. Iron granules found in those bacteria which metabolize iron compounds for obtaining energy. For example, iron oxidizing bacteria. Iron oxidizing bacteria. For example, Caulobacter, Gallionella, Sidrocapsa. Another example, it's about Equispirillum magnetotaceum contains magnetosome. This bacteria contains magnetosome that are vesicles having magnetic. The granules help the bacteria to orient themselves along geomagnetic lines. It's rare. C. Food reserve, yes. In past slides, I spoke about inclusion bodies, 1 gas vacuoles, 2 inorganic inclusions, and 3 reserves, yes, are absent. Natural fats are absent. Instead, polybeta hydroxybutyrate or PBH granules are present. Yes, in bacteria, we don't have any lipids as food preserve. Some bacteria have lipid. For example, Azotobacter. Azotobacter have lipids, but the type of their lipid is useful for plants. Protein granules are present. Carboxysome occur in photosynthetic forms. Another structure in cytoplasm, yes. For example, mesosome. Two types of mesosome, septal mesosome, lateral mesosome. Just about mesosome. It is a characteristic circular to villi form, a specialization of cell membrane of bacteria that develops as an ingrowth from the plasma membrane. Mesosome related to plasma membrane. It consists of vesicles, tubules, and lamella. Yes, vesicles, tubules, and lamella. Mesosome is 2 types, 1 septal, 2 lateral. What is septal mesosome? What is lateral mesosome? Septal mesosome. Septal mesosome connects nucleoid with plasma membrane. Yes. nucleoid DNA to plasma membrane connection. It takes part in replication, yes, replication of DNA, replication of nucleoid by providing points of attachment, the replicated ones. Septal mesosome is also believed to help in septum formation, yes, in cell division. In cell division, when a bacteria divided to two bacteria, it needs cells. It needs repair the cell wall. Yes, it is about septal mesosome. Septal formation in cell division related to septal mesosome. At the time of cell division, plasma membrane grows in the region where the septal mesosome is present, so that most probably it provides membranes for rapid elongation. This is the role of septal mesosome in cell division, yes, and in replication. Lateral mesosome. About lateral mesosome, it's not connected with nucleoid. Septal mesosome connected to nucleoid, but lateral mesosome, no. It's not connected with nucleoid. It contains respiratory enzymes. Yes, respiratory enzymes. It's important for metabolism. Respiratory enzymes, and is therefore often called chondroid. It is believed to be equal to mitochondrion of eukaryotes. Yes. In eukaryotes, in plant cell, in animal cell, in protozoa, in fungi, we have mitochondria, yes, mitochondria in respiration metabolism. But bacteria don't have mitochondria. Instead of that, bacteria have cell membrane, and mesosome in cytoplasm related to cell membrane, responsible for respiratory enzyme. However, respiratory enzymes are also present over the plasma membrane. This is a structure of mesosome. You can see cell wall, thick layer, cell wall, and then thin layer, plasma membrane, and mesosome. Yes, form of mesosome. And ribosome. Be patient. 2 and 3 slide. The class is finished. I don't know, you are tired. Excuse me. Ribosomes. Another structure in cytoplasm is ribosomes. You know, ribosomes involved in protein synthesis in eukaryotic cells and in prokaryotic cells. Yes, ribosomes. But the structure of ribosomes in eukaryote and prokaryotic cells is different. Ribosomes are small membrane-less submicroscopic ribonucleoprotein entities having size of 2 nanometer to 40 to 50 nanometer. Ribosomes are two types. Two types of ribosomes. One fixed and another free. Yes, fixed ribosomes free ribosome. Ribosome are attached to plasma membrane. Yes. Fixed ribosome attached to plasma membrane. But free ribosome occur free in the cytoplasmic matrix. Fixed ribosome is abundant, but free ribosome is free in cytoplasm. Ribosome Yes. Free or matrix ribosome synthesis protein. Do you have my voice? Do you have my voice? Yes? Yes, I continue. Yes, I continue. I think the time of class is out. But I think I can continue. Yes. Each ribosomes have two subunits, larger 15 subunits and smaller 30 subunits. Ribosomes take part in protein synthesis. Free or matrix ribosomes synthesize proteins for intracellular use, while fixed ribosomes synthesize protein for transport to outside. Yes? Free ribosome are important in synthesis of protein for intracellular use, but fixed ribosomes synthesize protein for transport to outside. Ribosomes generally occur in helical group called polyribosomes or polysomes. Many of ribosomes form polyribosomes or polysomes. In each polysomes, 4 to 8 ribosomes are attached to a single strand of messenger RNA or mRNA. It is a mechanism to synthesize several copies of the same protein. 3. Chromatophore. They are internal membrane systems of photosynthetic forms which possess photosynthetic pigments. Chromatophore is related to pigments of bacteria. It's related to photosynthetic bacteria because most of photosynthetic bacteria are pigmented. For example, in purple bacteria, purple pigmented. In purple bacteria, the membranes are typical, while in green bacteria, they are non-unit, non-lipid, and proteinous. Chromatophores of green algae contain photosynthetic pigments are bacteriochlorophyll, bacteriophytin or bacterioviridin, and carotenoids. Some of chromatophore in algae are carotenoids, bacterioviridin, and bacteriochlorophyll.